Rolling.
This lecture is about photosynthesis and chloroplasts. That's the sun. The Earth is very, very old. The Big Bang occurred about 15 billion years ago. The Earth has been around for almost a third of the life of the universe. The Earth is very, very old. Let's magnify the time that the Earth has been around and look at the timeline of a number of events in the evolution of the planet. Life. For a long time, there was nothing. The Earth was very, very hot. Then we had oceans and continents, and somewhere in there, a bunch of nucleic acids got together and formed this thing that people call the RNA world. RNA is very old. The first living cells started from precursors made in the RNA world. And sometime after that, about 3.5 billion years ago, that's a B, billion, the first photosynthetic cells formed. And this is amazing. I'm going to tell you how amazing this is. And these are kind of bacteria-like things. Bacteria. Relatives of modern bacteria. And the sun shone, and photosynthesis produces oxygen. Oxygen was soaked up by iron in the ocean to form ferric oxide for this long, for 2 billion years. It took 2 billion years before all of the iron in the ocean, which was dissolved, to precipitate in ferric oxide. When all that iron was precipitated, there's a whole bunch of iron on the bottom of the ocean. Then Earth's atmosphere started accumulating oxygen. Photosynthesis is responsible for all of the oxygen in the world. Right about this time, about 1.7 billion years ago, eukaryotic cells started to emerge, and chloroplasts started contributing to photosynthesis. Then we had plants, and then the Earth was colonized. And then somewhere here there's dinosaurs and human beings, and then lecturers like me. So let's boil it all down in a nutshell. Photosynthesis comes from the sun. That is, oxygen is produced and carbon dioxide is taken from the atmosphere, combined with water, to make sugars. Carbon from the atmosphere is making sugars. That allows people like me and people like you to use those sugars to breathe that oxygen, to pee, and to make useful chemical bonds. That's called respiration, and that's done by mitochondria, which we'll discuss in the mitochondria lecture. Chloroplasts, which are in plants, are related to those ancient bacteria, cyanobacteria. They're thought to be taken up in an endosymbiotic relationship with these cells. So most of this lecture is about energy. But there's also an information component to consider, because chloroplasts, like mitochondria, have their own genome. They have DNA because they're derived from bacteria. But the cell has to communicate with the chloroplasts to make all of the proteins. And in fact, most of the chloroplast protein, most of the chloroplast genes are encoded in the nuclear DNA now. That's the same with mitochondria. There's just a few left that are actually encoded by these organelles, plastids, mitochondria and chloroplasts. So, what do I want you to appreciate? Let's appreciate the sun and plants, because they're good. Without them, we would have no oxygen. We would have no life. And then we have to think about thermodynamics, because life follows the rules of thermodynamics. So I'm going to talk about energy flow. And it's important to compare chloroplasts and mitochondria because they work kind of similarly. So glucose is broken down by mitochondria and cells, and the free energy is negative 686 kilocalories per mole. That's a lot of energy, and delta G is negative, so the reaction goes forward spontaneously. Glucose is broken down in a series of small steps. So in order to make glucose, you've got to get that free energy from somewhere, right? Where are you going to get it? See if you can guess. So in mitochondria, glucose, or sugar, takes oxygen and is broken down into a number of intermediates to harvest the maximum amount of energy. And all of these energy carrier intermediates are used to make biological molecules and to carry out the reactions of the cell. If you burn it, all the energy would be released as heat. So it's a very efficient process the way cells do it. So that could be described as a controlled stepwise oxidation of sugars to efficiently harvest energy into energy carrier molecules. So if we turn that upside down, we have to think of building glucose, harvesting energy from the sun in small steps. So we have to have a stepwise elevation of electrons from chlorophyll to make NADPH and ATP for something that could be called anabolic oxidative phosphorylation. Isn't that a nice phrase? Anabolic means making stuff. Oxidative phosphorylation, it's just like in mitochondria. You've got the protons going across the membranes and this little motor thing spinning around making ATP. Pretty cool. So photosynthesis occurs in chloroplasts. Chloroplasts transform light energy into chemical energy using redox potential, just like mitochondria use that to harvest energy. They form a proton electrochemical gradient to make ATP, and then they use the energy carrier molecules NADPH and ATP to fix carbon from carbon dioxide from the atmosphere to make sugars. So mitochondria are small in relation to chloroplasts, and chloroplasts have an extra membrane. They have an outer membrane and an inner membrane, and inside that they have these things called thylakoid membranes. And inside the thylakoid membrane, it's very acidic. So let's look at the anatomy of chloroplasts. Just like in mitochondria, the outer membrane is permeable to small molecules and ions because of these barrel-shaped proteins which are in the membrane. And they look like a barrel, and they've got holes in them, and so the ions can flow through them rather easily. The inner membrane has transport proteins. It doesn't have those porins. So gases, they're nonpolar, so they can go across the lipid bilayer. They don't eschew water, and water diffuses at some rate. There's actually water channels as well. But transport proteins are required for things like ADP, ATP, and so on, for most solutes. That's the inner membrane, okay? So that's like the mitochondrial inner membrane. And now we're going to turn to these guys. Stacks are called granums, and these are called thylakoids. And these are the innermost membranes. These are inside the inner membrane. So they have a stroma, which contains all the carbon fixation enzymes, and that's not the membrane stuff. These are in granules, big protein machines in the stroma. They have DNA, they have their own genome, and they can replicate and they can translate, just like bacteria. And then they have these thylakoids. These are the membrane sacs, discs, that contain chlorophyll. This is where photosynthesis occurs. Okay, so photosynthesis in words. I'm going to show you pictures. Here it is in words. Light energy captured by chlorophyll drives the transfer of electrons and hydrogen across the membrane to form NADPH, the energy carrier intermediate. Protons are transported across the membrane into the thylakoid space. The inside is then very acidic, has a low pH. So that makes a proton gradient, which gets to be used to make ATP with the little motor thing, the ATP synthase. And then you got the high energy carrier intermediates. What are you going to do with those? Hey, let's make sugars. We'll fix some carbon. So that's how it looks in words. Now I'll show you some pictures. So chlorophyll looks like this. It's got a hydrophobic tail, so it can sit in the membrane. And it's got this porphyrin ring. And all these double bond, single bond, double bond, all these things, these are aromatic hydrocarbons, really. It's got this magnesium ion in the middle. These are really good at absorbing light. And so light absorption from this captures the energy and transfers it into a high energy electron. So it elevates the energy of this electron. So the light contributes to formation of intermediates, just like mitochondria, which pump protons and make these carrier molecules so that carbon fixation can occur. NADPH is kind of like NADH that we talked about with mitochondria. The difference is that NADH is used for catabolism, that is breaking stuff down, and NADPH is used for anabolism, that's making stuff. Okay? And the only difference between NADH and NADPH is this phosphate. P for phosphate. There's just a phosphate on it. Same molecule. Okay? But that phosphate allows it to be recognized by different molecules in the cell. So photosynthesis occurs in these things called photosystems. And these are amazing. They're kind of like dishes, right? They've got chlorophyll molecules in this kind of outer ring, and then they've got these inner sanctum chlorophyll molecules. You can call them the queen chlorophylls. So what happens is the chlorophylls in this outer dish part collect a bunch of light energy, collect a bunch of high energy electrons, transfer those high energy electrons into the central region that contains that queen chlorophyll. And then the queen chlorophyll is able to transfer that electron to a high energy intermediate, such as the quinone in the first reaction. So quinone is a molecule like used in mitochondria, just a little different quinone, but the same principle. Got the double bonded oxygens on two ends. So this diagram shows one way to think about how those electrons get converted into high energy intermediates. So the diagram shows these little balls here. These are supposed to be electrons. And when they go up here, that means they're going to a higher state. That means they have more potential energy, if you will. Kind of like, you know, higher to lower, right? Oops, I shouldn't drop that. We can edit that out. So the high energy, light energy excites the chlorophylls, and this queen chlorophyll eventually is the recipient of the final light energy. The queen chlorophyll then transfers this high energy electron to another acceptor, a high energy electron acceptor protein. And to replace that electron, this guy donates his low energy electron, and so now he's got a charge. So this guy's got a, he's reduced, he's got a negative charge. This guy's oxidized, he's got a positive charge. And then you've got to have another electron to fill that void. So a low energy electron comes in, and it comes in from water. So the first reaction center splits water. That's really hard. We'd love to be able to do that, to split water to make hydrogen so that we could burn it in cars. It takes a lot more energy to make hydrogen than you can get out of it from burning them. So photosynthesis can do that. There's a water splitting enzyme. And the water splitting enzyme takes that low energy electron from water, and guess what? Leaves a proton. Well, let's pump that into the thylakoid space. So you really get a lot of good stuff out of this water splitting enzyme. Then so this is all happy. We got this extra high energy electron. Okay, now we can put that into the electron transport chain. It goes to the quinone, the next guy. So water splitting results eventually from the absorption of energy from light. There's two photosystems. Photosystem 2 is the first one. That's got the water splitting enzyme. Photosystem 1 is the second one. Why are they called 2 and 1? This one was discovered first. So light is absorbed from the first reaction center. The water splitting enzyme releases 4 protons, and it releases oxygen. There's that oxygen. Thank you, photosystem 2. Transfers the energy to the quinone, plastoquinone. Then there's another electron transport chain intermediate, which also pumps protons, transfers the energy to plastocyanine. And then plastocyanine receives light and elevates its electron energy to get it further up in the potential energy diagram. And finally, ferredoxin receives the final high energy electron, and it gives it to this guy, which can transfer that high energy electron to make reduced NADPH. So, light comes in 2 steps. Each step elevates the energy. The energy of transferring from one to another goes down in between, but then gets elevated again on the second step. Why is that? You've got to get the energy of the electron high enough so that it can be put onto NADP to form NADPH. So the first step absorbs in the green, sort of bluish green, 608 nanometers. It releases 4 protons from the water splitting enzyme. And it transfers 2 electrons to plastoquinone. And so the net result is 4 photons plus water plus 2 of these quinones plus 4 protons gives you the transport of 4 protons into the lumen of the thylakoid space, which will be used later to make ATP, plus oxygen, plus the reduced quinone. this is a picture of the crystal structure of the reaction center that produces oxygen and splits water. So this is a remarkable thing. Without this protein, the water splitting enzyme, there would be no oxygen. Over billions of years, this guy is responsible for all of the oxygen in the Earth's atmosphere. After this guy works, we have quinone carrying the high energy electron to the cytochrome B6F complex, which also pumps protons as it transfers the high energy electron to the next intermediate. Then we have photosystem 1, the second guy, which takes that high energy electron and makes it even higher. Okay? We've got to get it high enough so that it can go down and add to NADP to form reduced NADPH. So light input transfers electron to then plasticyanine and ferredoxin, from plasticyanine to ferredoxin. And then ferredoxin NADP plus reductase forms NADPH, that high energy electron carrier that's used for formation of bonds with carbon, for fixing carbon. So the final step, 4 photons are required, and guess what? 2 protons are pumped into the thylakoid space again. So we're getting a lot of protons pumped, which we're going to use later. So this diagram explains it in terms of redox potential. Okay? So in this diagram, low energy electrons are elevated to higher states and then go down and then elevated again by photosystems. And remember that redox potential is basically the negative of the free energy. Okay? Negative of delta G. So this is a useful diagram. It shows that light energy takes the electron's energy up to this redox potential, where then it is transferred to the quinone. It goes down a little bit, goes down a little bit again, and then it's transferred to plasticyanine. So these protons are released during these transport steps, so that the protons are going into the thylakoid space to spin the motor to make ATP. So it's not a waste, it's transferring it to another form, transferring the energy to another form. And then, see, the problem is you got to get the high energy electron high enough in energy to form NADPH. And you can't get there in one step, that's the problem. So you got to do it in 2 steps. So photosystem 2 takes a slightly higher baseline, inputs light as energy, and then transfers it to ferredoxin and then ferredoxin NADP reductase to form that high energy intermediate. That's why it's a 2-step process. So then we have chemiosmotic coupling, just like in mitochondria. We have the ATP synthase on the thylakoid space, and ATP is made in the stroma. So you've got these discs, you've got the inside the thylakoid space, a whole bunch of protons. It's like pH 5. And then the proton gradient spins this motor, and it makes ATP, as I showed you in the movie on mitochondria. So chemists like to look at the energetic yield of things. And photosystem 2 takes 4 photons, 2 waters, and transfers those 4 protons. Try that again. Photosystem 2 takes 4 photons and 2 waters and some quinones, a couple of quinones, and transfers those protons to the lumen of the thylakoid space, releases oxygen, and you have reduced or high energy quinones. Those quinones then are used to pump more protons and regenerate the quinone. And then photosystem 1 takes 4 photons and makes our special 2 molecules of high energy electron carrier NADPH. So the result is it takes 8 photons to make 2 NADPHs. And then, in the meanwhile, you've got 8 protons translocated, which are then used to spin the motor and make, it takes about 3 protons to make 1 ATP. So the net result is we have enough energy to make glucose from carbon. How are we going to do that? We got NADPH, we got ATP. How are you going to get that carbon from the atmosphere? There goes some. Into sugars. Well, carbon fixation knows how to do that. So this occurs in the stroma of chloroplasts, and there's a lot of reactions, and I'm not going to go through all the details. But I'll tell you that it starts with 3 carbon sugars, and there's a bunch of oxidation reduction reactions, and the input is ATP for the high energy phosphate bond energy, and NADPH for those high energy electrons. So this is called carbon fixation, or the Calvin cycle, after this guy Calvin, who, can you imagine the work, discovered this process. So the first step is to take 3 molecules of carbon dioxide and add them to 3 molecules of this 5-carbon intermediate to form 6 molecules of this 3-carbon intermediate. And that's carried out by the most abundant enzyme on the planet, ribulose bisphosphate carboxylase, or Rubisco for short. The word on the street about Rubisco: sluggish but abundant. So here's what he does. Takes this carbon dioxide, adds it to this carbon. Here it is, carbon dioxide. He's painted pink here. And then splits into 2 molecules, forming 3-phosphoglycerate. So these can then be used as building blocks to make a number of things. So then use up ATP to put phosphate groups on there. The phosphates are high energy intermediates, and then you use up your NADPH to make glyceraldehyde 3-phosphate. Glyceraldehyde 3-phosphate is an aldehyde, so it's reactive. It'll form a number of sugars and fatty acids and amino acids. So it looks like this. It's a very useful building block. Finally, we regenerate our 5-carbon intermediate in the last step, which requires another 3 molecules of ATP. So 3 molecules of carbon dioxide form 1 molecule of glyceraldehyde 3-phosphate. It takes 9 ATPs and 6 NADPHs. So that's a lot of energy. But look, you're taking it from the air. I mean, what do you want? And then you get this guy, and he can be used to make starch, sucrose. Plants use sucrose. Sucrose is glucose linked to fructose. And it's transported in plants for energy. And it's also useful for making amino acids and fatty acids. So the net result from photosynthesis is that we have 6 CO2s from the atmosphere, 6 waters plus light, and that gives us sugars. This is glucose, plus oxygen. So let's thank the sun. Thank you.